Claire and I's project is on tree tumors and the bacteria that causes them, known as Agrobacterium tumefaciens. Agrobacterium tumefaciens is the main cause of crown gall disease or tree tumors in over 140 types of plants. The bacteria inserts a section of its Ti plasmid, which is the main plasmid of this type of bacteria, into the plant cell. This section of the plasmid is known as the tDNA and causes the plant cells to grow and divide rapidly. Agrobacterium tumefaciens is the main bacteria that causes tree tumors, but in our experiments, we are wondering if all of the tree tumors we find will be caused by this bacteria, or if there is another prevalent bacteria that causes them to grow.
For our first experiment, we're going to collect multiple samples from the first tree tumor that we find using a drill bit. We're going to test these samples in different types of media so we can figure out which strand of agrobacterium we have in the tumor. We're thinking we're going to use mannitol glutamate agar, but we're still going to test other medias just to make sure. After seeing which media the bacteria grows on, we're going to grow multiple colonies and then place the colonies on a plant such as tomato or carrot because they will grow the tumor easily. When growing the tumor, you can see on this timeline that the tumors grow pretty quickly. We're hoping that the tumor will grow within 2 weeks, and each day we'll take observations on the tumor growth. After we do this, our next hope is to see if the tree tumors have any similarity to human tumors. By doing this, we could take possible treatments or ideas that people have previously thought to work on human tumors and try to apply them to our tree tumors. There's not much of a budget for this project, but we are going to need carrots or tomatoes to grow our colonies on, and right now our drill bit's working, but in case it breaks or doesn't work when we actually get to the tree, we might need to buy another drill bit.
Over the course of this year, we would like to conduct the following experiments and possibly more. 1. Are all tumors caused by the same bacteria? 2. Do these tumors have any similarity to human tumors? And 3. Are there any possible treatments to prevent tumor growth on these plants? Our research might change depending on what we find throughout the year, but for now, this is what we are planning on.
This weekend, the 25th, we're planning to go on our nature walk to find and collect samples of our tumors.
Then next week, we're going to make different medias and incubate the samples that we got on these medias. And then by the end of the week, we're hoping to observe the growth of these colonies and choose which media we're going to use.
The week to follow that, we're going to collect more tree tumor samples and grow them on the media that we decided.
And then following that week, we're going to place the colonies that we grew on plants, either carrots or tomatoes, and observe the tumor growth on them.
From that point on, we're going to see what we found so far and where we think we can go from there.
Thanks for watching!